A gene is the basic unit of hereditary information that is passed from parent to offspring. Genes are made up of sequences of DNA arranged in a specific order with specific locations on the chromosome in the nucleus of the cell. The expression of a gene controls the synthesis of enzymes within a reaction. Enzymes help to catalyze reactions to produce, transform, or disrupt a chemical reaction or metabolism, specifically within our bodies. Enzymes are used in the manufacturing of medicines like drugs and antibiotics, for example, steroids. Gregor Mendel, the father of genetics and the famous pea plant scientist, conducted research consisting of pea plant genomics. He was able to discover that genes are found within the chromosome, and that specific genes led to specific phenotypical outcomes and patterns of inheritance through generations. Mendel cross-pollinated pea plants based on their different features to figure out which features were dominant versus recessive and how these traits were inherited between generations. In the first image, it shows the different characteristics that he looked at, or different traits. He first analyzed round versus wrinkled, then yellow versus green for the seeds. He also looked at purple flower color versus white flower color, pod shape, so inflated versus constricted, as well as yellow versus green color of the pod shape. Biochemistry aims to explain life at the molecular level by drawing together chemistry, genetics, biology at the cellular level to paint an overall picture and to make connections. The chemistry involved includes atoms like carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, which are all vital in our living bodily systems. These essential atoms are involved in the production of proteins and nucleic acids, which make up our systems we depend on, as well as our DNA. There are specific non-covalent interactions occurring within our systems, bonding DNA strands together to eventually form larger macromolecules. Non-covalent bonding does not involve the sharing of electrons. Instead, the two strands of DNA, for example, are held together by hydrogen bonding and form the double helix, and thus formations occur in the primary, secondary, tertiary, quaternary structures. An example of non-covalent interactions would be sodium fluoride. The positive charge of the sodium ion is attracted to the negative charge of the fluoride ion, but this bond can be easily broken by adding water or another highly polar solvent. Other examples of non-covalent interactions would be van der Waals, which has an attractive repulsive force, as well as dipole-dipole or ion-ion interactions, where there's also regular covalent bonding. Water plays an important role in our biological systems and processes. Water forms hydrogen bonds with other molecules and acts as a solvent due to its tendency to form hydrogen bonds and its polarity. For example, it is recommended to have an increased water intake due to water's ability to allow blood to flow through blood vessels more easily and avoid clotting of the blood within the bloodstream, as well as an increase in blood flow to organs like the heart, the kidneys, and the brain. Water also helps to eliminate any excess electrolytes your body may not be using, like sodium, potassium, and urea. Living cells operate under homeostatic conditions that are not at equilibrium. This requires the use of molar concentrations of substances that approximate the activities of solutes within a reaction. The Gibbs free energy equation is used to express this idea, showing that delta G is equal to delta H minus T times delta S, where delta G is the Gibbs free energy, delta H is the change in enthalpy, delta S is the change in entropy or disorder of that system, and T is the temperature of that system. The free energy equation is in reference to the second law of thermodynamics, where temperature is the determining factor whether the heat is being absorbed or released to induce a chemical reaction or because of the chemical reaction. When delta G is at 0, the system is said to be at equilibrium. Living cells operate at conditions that are not at equilibrium, so for many bodily systems, delta G does not equal 0. This is where RT times ln(q) describes the concentrations of reactants and the products. This is important in the medical realm when talking about diffusion across the concentration gradients. For example, dialysis or nerve conduction relies on the rapid movements of sodium and potassium ions through a concentration gradient. We can see in the bottom right picture, we have two tubules, an outside and inside tubule, showing that sodium is being pumped against the concentration gradient to get to the other side, as well as water diffusing down the concentration gradient. When it comes to nucleic acids, we have DNA and RNA. DNA stands for deoxyribonucleic acids, whereas RNA is ribonucleic acids. RNA consists of an OH group attached to its sugar, whereas DNA only has a hydrogen at the end of its sugar instead of an OH group. Both consist of phosphodiester links and the DNA or RNA ring, as well as the base in the 5' to 3' direction always. Bases can either be purines, which would be your A and your G, your adenine and your guanine, or your pyrimidines, which would be your C, T, or U for RNA. This would be your cytosine, thymine, and uracil. Our genetic information is coded within the double-stranded DNA. This is the genome. The genome of a virus, though, is slightly different because it can be encoded within a single strand of DNA or even the RNA of the virus. What a cell is like or consists of and what the cell can do as far as function goes is all written within the DNA sequence and what proteins that sequence formed. Someone's genome can be transcribed and evaluated to see if there are any mutations occurring that could be the cause or the outcome of a disease or illness. That top photo is showing the DNA structure, very basic form, going from the 5' to 3' direction, and then that bottom strand is going the opposite direction with its complementary base pairs. Our bottom photo shows the central dogma, so how DNA goes to RNA, goes to a protein to create that polypeptide chain of amino acids. This is because of the transcription that occurs to create the RNA strand, and then that is translated into our polypeptide amino acid chain. Proteins are polymers of amino acids that are made up of 3-letter codes or codons. There are 22 amino acids that can be encoded within your DNA. There are sequences that to indicate start and stop with a specific codon, AUG being a start codon, UAA, UAG, or UGA being the possible stop codons for that sequence. These protein sequences are what allow for genome sequencing to determine genetic abnormalities or illnesses. Someone in the lab may decode this genome and look at these 3-letter codes to figure out what it's coding for. If we look at that middle picture, this shows you where that AUG start codon is occurring and how that AUG is translating to methionine, the MET, at the N terminus of the 5' end of that DNA strand. The C terminus is towards the 3' end of that DNA strand. This one shows the stop codon of UAA for that polypeptide. Protein sequencing is important when it comes to mapping evolution and proposing evolutionary relationships or creating a phylogenetic tree. Species with a similar protein sequence are likely to have a common ancestor that they share the gene sequence with. Although sequence similarities can still arise without a common ancestor, these discoveries help us further conclude how protein structure and function is related, and how that can affect us and the way we function. This is why genetic testing is done during pregnancy in order to get ahead and see what can be done if necessary to help or fix a mutated gene. Protein sequencing plays a vital role in drug discovery and development by allowing us to design molecules like biologics that can interact with these proteins. Protein molecules have different levels of organization. There are primary, secondary, tertiary, and quaternary structures, all of which results from the non-folding and folding of the protein sequence determined by the gene sequence. Whether the protein continues to go through the folding process to the quaternary structure can determine its role in a system. Issues can arise within a system if a protein is inappropriately folded or misfolded, changing the structure of that protein and then further limiting its ability to play its correct role in the system. The primary structure consists of a linear chain of amino acids, whereas the secondary structure contains regions of amino acid chains that are stabilized by hydrogen bonds from the polypeptide backbone. These hydrogen bonds create the alpha helix and beta pleated sheets. The tertiary structure is the 3D shape of the protein, and that is determined by the interactions of side chains from the polypeptide backbone. The quaternary structure influences the 3D shape of the protein and is formed between 2 or more polypeptides, creating a tighter knit structure. It kind of shows it on the picture to the left. you can see how it's just getting more closely tight knit together. That primary structure is just that straight chain of amino acids. That secondary is creating that alpha helix shape. Tertiary is getting a little bit folded and messy, and then quaternary is that those multiple polypeptides all forming together. The proper folding of a protein helps to stabilize the protein and allow for the correct bonds to occur. Hydroxylation of proline and lysine occurs to form stable bonds, but requires vitamin C for the enzymes. When someone has a vitamin C deficiency, this can lead to a failure of hydroxylation to occur, specifically with collagen fibers, thus reducing the hydrogen bonds on the protein sequence. Vitamin C is essential for proper protein function. This can reduce oxidative protein damage, which can lead to cell death. This is where you may hear a lot about the benefits of antioxidants due to the connections with ROS, or the reactive oxygen species. If you look at the bottom image, you can see that proline was hydroxylated to the hydroxyproline, the addition of that OH group. And then just some signs of vitamin C deficiency, which is important to just be aware of, would be dry or splitting of your hair, dry skin, nosebleeds, joint pain, easily bruising, immune system impairment. That's usually a very easy one to spot. And that's why a lot of people like to take those vitamin C little packets